So hello everyone, welcome back to today's class. In today's class, I'll be talking about the cell organelles. This class is in continuation with my previous class. In the previous class, I have already spoken to you about the history of cell. Okay, I've spoken to you about who discovered cell and then how the cell structure is, and what is the smallest and the largest cell. Okay, about prokaryotic and eukaryotic cell, also about plant and animal cell, and the functions of a cell, about cell theory. So in today's class, we'll continue with that, with the previous class, and we'll talk about the cell organelles, some of the cell organelles. Okay, so let's start with today's topic. So in today, we'll talk about the cell wall, cell membrane, cytoplasm. Okay, before we go that, let's talk about what are the components present in a cell. So in a cell, there is the cell wall is present. Okay, cell membrane is present. Okay, now remember, cell wall is present only in plant cells, not in animal cells. Okay, so cell wall is present, and then outer part is cell wall, and then we come to the cell membrane. Then we get cytoplasm. Okay, then cytoplasm is a liquid fluid part in which all the organelles are present. Okay, so cytoplasm, it helps in the maintaining the transport of, it helps in transport of substances or molecules or materials inside the cell. Okay, it also holds all the organelles inside the cell. Then we come to mitochondria is present, nucleus, endoplasmic reticulum, Golgi bodies, ribosome, plastids, vacuole, lysosome, centrosome, cilia and flagella, and cytoskeleton. Okay, there are more so we'll talk about all this components of a cell. So in today we'll talk about, first let's start with the cell wall, cell membrane, and cytoplasm. So cell membrane, it was explained by Singer and Nicholson in 1972, and they gave the fluid mosaic model of cell membrane. So I'll discuss about this in detail in my next slide. So just remember this, that in human beings, these are some small points which you can learn, which you can learn for your medical exams and all. So the membrane of the erythrocyte, okay, has approximately 52% protein and 40% of lipids. See, basically, cell membrane, it contains lipids and proteins. Okay, 50% lipids and 50% proteins. So in human beings, if we talk about human beings, then the erythrocyte membrane, it contains 52% protein and 40% lipids. Now, there's a topic known as membrane fluidity. Okay, so plasma membrane, or you can say cell membrane, it maintains this membrane fluidity. Okay, it helps in maintaining the fluidity of the cell. So what is the function of that fluidity? So it helps in the cell growth, formation of intercellular junctions, and secretion, endocytosis, and cell division. Okay, now there's another topic, another point, or topic you can say, known as the active and passive transport. So basically, when this cell membrane, okay, when the materials, when they are transported in the cell membrane, then there are two type of transport we talk about. One is the active and one is the passive. So when the transport is occurring without without using ATP or without using any energy, it's a passive transport. Okay, and when the transport is occurring with using the ATP or against the concentration gradient, then it's known as the active transport. Okay, remember, two type of transport is present when we talk about transport of molecules in and out of the cell membrane. So active and passive transport. Okay, now let's move on to the next slide, where we talk about the fluid mosaic model. I've already told you that G.L. Nicholson and S.J. Singer in 1972 gave this model. Okay, so let's talk about the model, what is present in this cell membrane and all. Okay, so now you can see out here, first we'll talk about the membrane proteins. See, it's very important. Okay, first of all, cell membrane, it contains lipid and protein. Remember this. Okay, it contains lipid and protein. Also, it contains carbohydrates. Okay, so basically, it's a mixture of lipid, protein, and carbohydrates. So this phospholipids, okay, you can see out here, phospholipids. This is a major lipid present in the cell membrane. Okay, and there are two membrane proteins known as the peripheral membrane protein and internal membrane protein. Remember, the peripheral membrane protein, it lies towards outside the lipid bilayer. Okay, basically cell membrane is a phospholipid bilayer. So you can see two layers are there. This one layer, this one layer. Okay, this two bilayer. It's a phospholipid bilayer. Okay, and inside you can see there are you can see this you can see this part. This is the hydrophobic part, and this is the hydrophilic part. Sorry, hydrophilic part is the outer part, and this is the hydrophobic part. You can see polar heads out here. So since they are out towards the outer part, so they are hydrophilic, and the tails which are inside toward facing each other, this is the hydrophobic part. Okay, so they hydrophobic means what? They do not love water, they fear water, and hydrophilic is water loving. Okay, so the heads are outside and the tails are inside. Okay, now membrane protein, there are two membrane proteins. Peripheral membrane protein, this towards the outside, okay, outer part of the phospholipid bilayer, and integral protein, it is present in the inside part, okay, inside of the phospholipid bilayer. Okay, this to remember this too. Then cholesterol is also present. Cholesterol, what does it do? The amount is almost same like the phospholipid bilayer, and cholesterol is helpful for maintaining this membrane fluidity. Okay, so for transportation, for maintaining the structure of the phospholipid bilayer and all, cholesterol is helpful also for membrane fluidity. So now there are some, okay, now since this contains phospholipid, okay, lipids and proteins, so remember, the molecules which are water soluble, they cannot pass through this cell membrane. Okay, to pass through the cell membrane, they need either carriers or they need channels or pumps. Okay, you can see a protein channels, and there are also pumps, and also they need carriers. With the help of those things, this water soluble molecules, they can uh, they can move across the membrane. Okay, so just remember that. Now you can see a glycolipid out here. This lipid is attached with the carbohydrate. Carbohydrate, they are present on the surface of the phospholipid bilayer, which attaches with the lipid, forming the glycolipid. Also, when carbohydrates stay attached with the protein part, then they are known as the glycoprotein. Okay, so this is glycoprotein, glycolipid, and this I've told you about the things and all. Okay, now you can see a cytoskeletal filaments. What does it do? The cytoskeletal filament, it helps in the shaping of the plasma membrane. Sometimes the plasma membrane, the shape has to change. Sometimes for movement, or for movement, yeah, for movement or for different purpose or functions, this plasma membrane, it has to change its shape. So for that, the cytoskeletal filaments, they help. Okay, so this is all about the cell membrane. Now, moving on to the next part, cell wall. So cell wall, basically it contains three. You can see out here three layers are there in cell wall. So what is the three layers? Middle lamella is present. Okay, primary cell wall and plasma membrane. So we know that cell wall is the outer part. So outside cell wall, remember there is another part known as glycocalyx. So after that we have the cell wall. Cell wall is the middle lamella and cell wall, primary cell wall. Okay, and inside part it is the cell membrane. So cell wall is present only in plant cells, not in animal cells, and basically in cell wall you can see what is there. The network of cellulose microfibrils, okay, which is embedded inside the network of pectin and hemicellulose. You can see here, see, pectin and hemicellulose. So inside it, the cellulose microfibril is present. Okay, so now middle lamella, it is composed of calcium pectin. Okay, what does it do? It holds as we know that cell wall is the outer part, so it helps to hold one cell wall, the cell wall, with the neighboring cells. Okay, and what does the cell wall in what does the cell wall do? So cell wall, it provides shape to the cell. Okay, it also protects the cell from any mechanical damage and infection. Also, it helps in the cell to cell interaction, and also provides barrier to undesirable macromolecules. Okay, so kindly bear with me. I'm suffering from a bit cold. So moving on to the next part. So I hope you have understood cell wall. Okay, cell wall function, I've told you. Basically, the cell wall is the outer part, right? So it provides shape to the cell and also protects the cell from the mechanical damage and infection. Remember this. Okay, it also protects the cell by not allowing toxic chemicals or molecules to enter the cell. Okay, remember this thing. So now in mitochondria, next is mitochondria, very very important. Mitochondria is also known as the powerhouse of the cell. Okay, why? Because it helps in the formation of ATP or energy. Okay, so it produces cellular energy in the form of ATP. So it is known as the powerhouse of the cell. Remember one more thing, mitochondria is also a semi-autonomous organelle, which because it contains its own DNA. Remember this, it contains its own DNA, okay, and it also produces energy. So it's also known as semi-autonomous organelle. Remember this, okay. Another one is chloroplast, other than mitochondria, which I'll talk to you about that later, okay. So let's come back to mitochondria. So in mitochondria, you can see here, see the structure of mitochondria out here. See, DNA is present inside. Okay, it contains its own DNA. Also, it contains ribosomes. So, ribosomes basically they help in protein synthesis. Therefore, since it contains ribosome, it can also synthesize proteins. Remember. Now you can see this folding part. Okay, this folding part. So, what is this? Is known as the cristae. Okay, so cristae, what does it do? It increases the surface area. Okay, and it extends to the part known as the matrix. Internal portion is known as the matrix. Okay, so it contains outer membrane and inner membrane. Okay, then the inner membrane it contains, you can see outer F0 and F1 portion. So basically this helps in the uh, final process of ATP production. Remember, when energy is being produced, ATP is being produced. These two portions they are helping in the final steps of ATP synthesis. Remember that, okay? So this is the intermembrane space. The space between the outer and inner membrane as well as the intermembrane space. Fine. Now, what is the shape? It looks like a cylindrical shape, and the diameter is 0.2 to 1.0 micrometer. Fine. And the length is around 1 to 4.1 micrometer. Remember that. Okay. Now, what are the functions? I've already told you. It helps in production of ATP. Okay. These are sites of aerobic respiration. So out here, aerobic respiration takes place. Okay. Now, they possess their own singular circular DNA. They contain RNA molecules also, and the ribosome says 70S. Remember, the subunit is 70S. Okay, and since they contain ribosomes, so they are also helping in the protein synthesis. Okay, now let's move on. Now nucleus. So nucleus is also very very important. Nucleus is taking part in reproduction process because it contains this DNA. It also helps in passing of uh, genetic information. See, nucleus contains DNA, and DNA is the heredity information. Okay, it is the genetic information. So basically, nucleus indirectly it is also taking part in this process of reproduction and also the passing of the genetic information from the parent to the offspring. Okay, so nucleus it contains a nuclear membrane. You can see here it contains a nuclear membrane outside. Okay, and then it there is okay. So this is also known as the nuclear envelope or nuclear membrane. So now there are pores are present. Okay, nuclear pores are present. So these are the passages. Okay, through which movement of RNA and protein molecules they take place. Okay, so from nucleus to cytoplasm, from cytoplasm to nucleus. So we know that there is a cell. Okay, and inside the cell there is the okay. This is the cell. This is the nucleus. So from cytoplasm, the molecules are going outside, and from cytoplasm, it's coming in, and from nucleus, it's going outside. Okay, so nucleus it contains a nuclear membrane which contains pores through which this transport of molecules is occurring, and also it contains nucleolus. See, nucleolus it is a dense, it is a dense dark colored structure present inside the nucleus. So nucleus it's an active site for it's a site for ribosomal RNA synthesis. Okay, rRNA, ribosomal RNA synthesis. This nucleus, remember this. Okay, so it also contains network-like structures, like dense network-like structures known as the chromatin network. Okay, so chromatin is basically it is in a condensed form. Okay, so it contains chromatin network. It contains nucleolus, which is a dark structure. Okay, it also contains nuclear pores through which in structure. If I show you, it's like this. Okay, it's like this. So these are the pores through which the transport is occurring. Then we have nucleolus, and then we have the chromatin structures. Okay, so these are all are present inside the nucleus. Remember this. So the nucleus was first described by Robert Brown in 1831. Okay, and also this chromatid, it was named by Fleming. Remember this. So this is all about the nucleus part. Moving on to the next part, that is the plastids. So remember one more thing, plastids are present only in plant cells, okay, or in animal cells, remember that. And in plastids, again, plastid is divided into, plastids may be of three types, that is the chloroplast, chromoplast, and leucoplast. So when the plastid contains chlorophyll, it is known as the chloroplast. When it contains xanthophyll or carotenoids, it's known as the chromoplast because they give the reddish orange color to the like they are responsible for this coloring of orange red color. Okay, so when they are present, xanthophylls and carotenoid pigments are present, then it's known as the chromoplast. So when only chlorophyll is present, which is resulting in the formation of green color, okay, so it is known as the chloroplast. Okay, and when there is no pigment, okay, neither green, red, orange, or whatever, it's known as the leucoplast, or you can say the colorless plastid is known as the leucoplast. So chloroplast, chromoplast, and leucoplast. Now again, leucoplast, there are three types. Okay, amyloplast, eleoplast, and aleuroplast. Okay, so leucoplast is of three types. Okay, amylo, elio, and eleuro. So when this leucoplast, when it contains carbohydrates, it is called amyloplast. Okay, when it contains lipids or fats, it's known as the elioplast. Okay, elioplast. When it contains proteins, it is known as the eleuroplast. See, I've given here the types of leucoplast. Okay, now how does the plastid look like? It's a oval shaped, or you can also say discoid shaped. Okay, this one. So now it's very important. See the structure. Out here you can see outer membrane is present, inner membrane is present. Now you can see some coil-like structures stacked together. So this is known as the thylakoid. Okay, if I show you like this, there are two membranes present. Okay, so when this coil-like structure is known as the thylakoid. So when it is stacked together, many thylakoids are stacked together, then it is known as the granum. Okay, this is known as the granum. But when it's single, it's known as the thylakoid. So when there are many stacked coil structures, they are attached, okay, by a thread-like structure known as the stroma lamelli. See here, stroma lamelli. Okay, and the inside fluid material is known as the stroma. Okay, the internal part is known as the stroma. So you can see the structure also out here. Okay, the thylakoid is filled with the space inside the thylakoid. It is known as the lumen. Remember this. Okay, so outer membrane, inner membrane, lumen is present inside the thylakoid. The thylakoid is present, and your stroma and stroma lamelli, and granum is the stack. Okay, so this is the structure of the plastid. Now what is the function out here? Remember one thing, that plastid is also a semi-autonomous organelle because it contains its own DNA. Okay, it contains its own DNA, and it also contains ribosomes. Okay, so since it contains ribosomes, it helps in the protein synthesis. And remember one thing, the ribosomes in chloroplast is 70S. Okay, and they are smaller than the cytoplasmic ribosomes. Ribosomes present in cytoplasm, they are of 80S subunit. Okay, and these ones present in plastids, it is 70S. Okay, and it contains small double-stranded circular DNA molecules and ribosomes. Okay, and out here carbohydrate and protein synthesis takes place in the plastid part. Okay, so remember this thing. Okay, now the amount or quantity of plastid in Chlamydomonas, it is 1. Okay, and in basophil cells, that is in leaves and all, there are 20 to 40 plastids present per cell. Remember this. Okay. Next is the endoplasmic reticulum. So this is also very, very important. This is the largest cell organelle, you can say. This is the largest cell organelle in the cell. And endoplasmic reticulum, what, how does it look? See, you can see out here, these are all sac-like structures, okay, all stacked together, and they're present outside the nucleus. Okay, it's present outside the nucleus. Now, they're of two types. Okay, they're of two types. One is the rough endoplasmic reticulum, and one is the smooth endoplasmic reticulum. So this is also known as RER, and this is SER. Okay, now why it is known as rough? Because when this endoplasmic reticulum, when it contains ribosomes, then it becomes rough. Okay, ribosomes are present on the outer surface of this endoplasmic reticulum, so it is known as the rough endoplasmic reticulum. And when ribosomes are absent, it's known as the smooth endoplasmic reticulum. Okay, so two types. So what are the functions? So rough one out here, it helps in protein synthesis. Okay, protein synthesis, and it's smooth, it helps in lipid synthesis. Fine. Also, the smooth one helps in calcium storage. Okay, it helps in calcium storage. Remember this. Okay, so these are basic functions of our endoplasmic reticulum. Also, see out here, when they are forming this proteins or lipids, they are again passed on to the Golgi apparatus for the transport of those uh, proteins to the uh, target sites. Okay, to the target places. Now you can see out here, these sac-like structures, they are known as the cisternae. Okay, they are known as the cisternae. Remember this. So this is all about the endoplasmic reticulum. Now moving on to the Golgi apparatus. So Golgi apparatus was given by Camillo Golgi. Okay, so Camillo Golgi, he discovered that he gave this Golgi apparatus. So again, same, Golgi apparatus is also, you can say out here, it's a sac-like structure. See, it's a sac-like structure like this. Now out here, Golgi apparatus, it contains two phase. What is this phase? What is the trans phase? Okay, trans phase is the maturing phase, and this phase is the, can say this is the forming phase. Okay, so outer what's happening is that when these proteins are being formed in the endoplasmic reticulum, these proteins, they are transferred to the Golgi apparatus. So out here what happens? Out here, packaging, modifying, and then transport of the proteins that are produced in the endoplasmic reticulum. Remember this, okay? Very, very important. Function of Golgi apparatus is the packaging, modifying, and then transport of the protein molecules to different target places. Like it may go to the cytoplasm, it may go to the lysozyme, or it may go outside of the cell also. Okay, so this is the function of Golgi apparatus. Remember this. Okay, and it's also a very important site where glycoproteins and glycolipids are formed. Okay, they're synthesized. So what happens? See out here, the proteins are coming here, they're packaged out here, modified, and then they're transported. Okay, they are transported. So when it comes towards this is the trans phase, okay, the maturing phase. When it comes here, then they're released. Okay, they're packaged, see, they're released. So this is a newly formed vesicle. So once it's packed and all, then they're secreted to the target sites or target places. Okay. So today, in today's class, we'll talk about Golgi apparatus. But next class, we'll be covering the rest organelles. So I hope you've understood in today's class. If there's any doubt, please do comment. And if you like the class, do press on the click icon, share it with your friends, and also subscribe to my channel for more videos. Okay. Please check my next class, part 3, and the finishing part of this topic, the cell unit of life. And thank you for attending today's class. Thank you so much.